who are new to Journal Club, and welcome back to everyone that's been here before. Before I start going back to the question thing, you can ask me any questions, but do remember that we need to be done by 8 o'clock. So if you think your question is going to be a little hard, you can email me, you can come up to me after. I'm more than happy to answer any questions that you want.
Sorry, I forgot one thing. IRT meeting next Tuesday morning at 7:30. Students. Sorry.
Also, for anyone who hasn't taken, for anyone who hasn't taken a biology course yet or hasn't taken Bio 2, if you don't understand anything, ask me to explain it. I would be more than happy to explain it in really, really simple terms. I told you that you didn't need to know science, which is kind of true. It is kind of true, I promise. So if you need me to explain anything, I'm more than happy to explain it to you. So before I start, I want to thank a couple of people. Mr. D, who has been Mr. D and me for a couple years now. But that comes with its perks and its bad things, and the perks are that I get to be here doing this, and it's a really big thing for me, and it's really important to me to be here. So thank you, Mr. D. Mark and Amanda, who have been the most amazing people to suffer with through this whole process. I love you guys so much, thank you, and both of you have had, like a huge impact on the way this club has gone. Ms. Legerifo, who answered, I think, 16 emails from me in the past 2 days over things that I didn't understand. So thank you for that. My entire Journal Club team, you guys have been amazing. Without you, this club wouldn't run, and I wouldn't be standing here. Dr. Cilio, who didn't specifically help me on this, but has been a huge part of Journal Club for the past, 3 years now? 3 years we've been doing this. She gets nearly every paper that you'll ever see presented on, so thank you for that. And my parents, Amy John, and my sister Dylan, for just, putting up with me being a huge science nerd all the time and getting frustrated and being really excited about stuff that they don't understand or don't really care about that much, but they're really supportive nonetheless. So with that, my paper is called Beta-catenin Mediates Stress Resilience through Dicer-1 MicroRNA Regulation. And normally we'd have a layman's title, but I don't think that's going to clear anything up on this one. So I think the first thing that we need to address is what exactly is stress. The dictionary defines stress as a state of mental or emotional strain or tension resulting from adverse or very demanding circumstances. So a situation that would be stressful is, in fact, angry. Anything that has to do with stress, you can probably relate to something back to the school, which is good and bad, because stress has some good aspects and some bad aspects. So some good aspects are what you would see in, say, animals, where there's a fight or flight response. So what the stress reaction does there is prepare the animals to either fight or flee. In other instances, which is chronic stress, stress can be bad.
I know it's coming.
So what chronic stress is a repeated stressor that happens a lot until the body can't really determine when it's happening versus when it's not happening because it's happening so often. And chronic stress has been linked to a couple of really bad problems such as cardiovascular failure, cancer, and viral flare-ups, which for anyone who knows what those are, you know they're pretty bad. So what my paper focuses specifically on is stress response and the nucleus accumbens. And before now, the nucleus accumbens has really only been linked to reward, pleasure, and motivation response. But what they found now is that the nucleus accumbens might actually play a role in stress through the Wnt signaling pathway. The Wnt signaling pathway is a pathway that is really prominent in cell development. It's tied together, and it's in almost every living organism, and is a huge, stepping stone for biology. Anyone who's a biologist, and anyone who's going to really study biology is going to know what this pathway is, and know that it plays a huge role in the cell. And what they realized, or what they observed in past studies, was that when mice are depressed, when there's impaired signaling of the Wnt pathway in the nucleus accumbens, so in just this part of the brain, the mice tend to be depressed. They've noticed that there's a relationship between the impaired signaling and the stress. So a huge part of the Wnt pathway is beta-catenin, which If you're looking here, here's wind, here's the start of the pathway, and then here's beta-catenin. So they're pretty close to each other in this pathway. Speaking more literally. So what they decided to look into was whether beta-catenin had an effect, like the different levels of beta-catenin in the nucleus accumbens had an effect on stress resilience, which is the ability to cope with stress. So I'm going to keep referring back to this diagram and adding to it throughout the presentation. The first thing you need to know is that wind activates beta-catenin. Remember that as we go through this. So the first thing that the scientists wanted to see was, like I said before, whether beta-catenin levels in the nucleus accumbens had an effect on stress resilience. So what they did was they went into the nucleus accumbens and injected a vector with beta-catenin in it or just a control vector. And they did this by using a herpes simplex virus, which was the vector. And then they performed 2 stress tests on the mice. The first one was the forced swim test, which is when they take a mouse and kind of just dump it in a tub of water and see how long it stays still versus it's swimming. And a mouse staying still in water means that it's stressed. That's an anxiety phenotype in mice. So as you can see, for the control versus the beta-catenin, the mice spent much more time immobile with the overexpression of beta-catenin than they did without it. Again, you can see the same results when they did the plus maze test. This is what an elevated plus maze is. There's 2 closed arms and 2 open arms, and a mouse spending time in the closed arms means they're stressed. So you can see that the mice spent significantly more time in the open arms when they had elevated levels of beta-catenin. So what they were able to conclude from this is that beta-catenin does have an effect on stress. So in the wind pathway, it is beta-catenin that is having this effect on stress. And more beta-catenin equals less stress, and it's the same way, the opposite way. So less beta-catenin equals more stress. Does anybody have any questions on the way I explain that? Do any new people have any questions on what I'm even talking about? Okay, so this is where this pathway goes to now. We have wind, the wind pathway. When there's elevated beta-catenin, there's less stress. But stress isn't a physical thing. There's not one thing that says stress. You don't have a gene in your DNA that says stress, and when that's produced, you become stressed. Stress is a bunch of different factors. So what they were looking for was what comes in between. So they did this by looking at different aspects of the wind pathway. So what they did was they subjected a group of mice to accelerated social defeat. They had control mice, susceptible mice, which are mice that don't deal well with chronic stress, and resilient mice, which is mice that do deal well with chronic stress. Does that make sense to everyone? So what they found was that in mice that were resilient, so mice that dealt well with chronic stress, 2 transcription factors that are downstream in the pathway from beta-catenin had elevated levels. So what that means is that beta-catenin, upregulation of beta-catenin upregulates these transcription factors. Yes? So what that led the scientists to believe was that these transcription factors are now upregulating something that they transcribe. So this purple question mark has now become TCF3 and TCF4, transcription factor 3 and transcription factor 4. But now we have a new question mark. If a transcription factor transcribed for something, what exactly is it? What is the missing piece in between what's the transcriber and what's happening, and stress? So they did this really complicated thing called genome mapping. And that's a little tough. You won't ever learn that in basic level biology courses here at Pingree. So we're going to skip over what exactly that is and just say this is what they found. You have to believe me, they found that these specific proteins are what TCF3 and TCF4 transcribe for. So what they did, again, was the same accelerated social defeat. I forgot to explain that before. What that is 4 days straight, they suggest, I don't remember the word. They subject, thank you. They subject the mice to twice daily social defeat. So they put so what they're doing is they're basically putting a little mouse in the cage with the bully mouse twice a day and letting the bully mouse bully the little mouse twice a day for 4 days. Yes?
So for stress tolerant versus non-stress tolerant, were they identifying those just based on observed behavior, or were there other?
I'm not exactly sure what they were doing that on, but I can look into that. I believe they did stuff in past studies that led them to differentiate and like be able to differentiate, but I can get back to you on that. So they did the accelerated social defeat, and again looked at the protein levels in the mice, in the different mice. So full control right here, the y-axis, is basically, in simple terms, how much of the protein is being expressed. And what they found was that Dicer1, which plays a really important role in destroying bad mRNA in your cells. So mRNA, so there's DNA, mRNA, simply DNA, mRNA, protein. DNA, the transcription factors, transcribe for mRNA. And mRNA is the template that the cell uses to make the protein that that DNA wants it to make. Does that make sense to all of my new people? Did that make sense to my old people? Because if it makes sense to them, then I'm good. Cool. So what Dicer1 does is basically destroys the bad stuff that the cell tells it to destroy. So that's really important, that they found something that they know plays a huge role in cells. Yes?
You said it destroys bad mRNA. What exactly is it? Just it's kind of transcribed.
One thing that would be considered bad mRNA, which is going to be a little viruses. Double-stranded RNA, mRNA, which would be a virus. For those of you who don't know that, don't worry about it. It really doesn't have that much to do with the paper. So what they found is that there were elevated levels of Dicer1 in the resilient mice, which led them to believe that TCF upregulation of beta-catenin upregulates TCF3 and TCF4, which leads to an upregulation of Dicer. So the next thing they did is a little complicated. They decided to take out Dicer and see what upregulation of beta-catenin does. So the way I'm going to explain this is explain this part and then go to here and show you which parts of this relate to this. So the first thing they did was inject the mice in the nucleus accumbens with either a control or the herpes simplex with this thing called Cre-Lox. And what Cre-Lox is a DNA recombinase. So basically it's what your cells use to cut and insert or cut and reattach DNA. So they did that here and then made the mice go through social interaction, which is what you see here. These are the amount of time they spent in the interaction zone in a social interaction scenario. So then after that, they injected the mice with the beta-catenin vector. So they upregulated beta-catenin in both the mice who did, these mice who did have the Dicer1 and these mice without the Dicer1. And then they subjected them to accelerated social defeat, so the same exact thing that they were getting before, or both the mice in the past mice have been getting. And then again, they watched them in social interaction, so social interaction post-accelerated defeat. And what they found is this right here. The mice with the elevated levels of beta-catenin and the Dicer1 had good effects. So after the social defeat, they still interacted more than the mice without the Dicer1. So this led the scientists to figure out that it is in fact Dicer1 that's having this effect on stress. It's not just beta-catenin, it's beta-catenin that upregulates Dicer1. So the point the scientists are trying to make with this is that you can't just have big parts. They're looking for the individual parts. These pathways that they're using have huge effects on cells. These proteins that they're looking into have a huge effect on the development of cells. And if you cut something out at the beginning, the things that happen downstream of that, so the things that that controls, could change. And sometimes those things have such huge implications on cell development and cell health that it could destroy the cells. So what they figured out is that this little purple, or I guess pink question mark here, has now become Dicer 1. Yes? Something really, really cool that they did in this study that a lot of times researchers don't have the opportunity to do was compare their findings with that of humans. So this is the graph that I showed before with the TCF3 and the TCF4. And remember, we were looking at that and we saw that when the mice were resilient to stress, they had elevated levels of TCF3 and TCF4. So it's a little different than what this is showing. This is showing that when they looked at the nucleus accumbens in humans post-mortem who knew they had depression, so they were diagnosed with depression and had depression at the time of their death, they found that there were down-regulated levels of TCF3 and TCF4. So that's kind of opposite, but they're showing the same thing, that when there's upregulated levels of TCF3 and TCF4, there's less stress, and when there's lower, there's more stress. So even though there were some slight differences, like here, axon 2 and axon 2, it's a little bit different and there's a couple inconsistencies. It's really important to show that these have a lot of similarities. So in conclusion, the main point of the paper is beta-catenin activates a network in the nucleus accumbens that downstream upregulates Dicer1. So if beta-catenin is upregulated, it upregulates Dicer1 behind it. And that's really important because we now know that Dicer1 is destroying something in the cell that has to do with stress. And it's also really important to take away from this that the human model and the mouse model may have had some inconsistencies, but the similarities are huge because, let's be real, nobody really cares if your mouse is stressed. The reason we're doing this is not to make stress pills for mice, it's to make maybe look into stress therapies and treatments in the future for humans. So what's next? The first thing that they need to look into, the next part of research that they should do, is figure out what Dicer1 actually is destroying. Because if you just knock out Dicer1 like they were doing, you are taking away the cell's main ability to destroy that bad template, that bad mRNA. So that would be the next thing they have to look into research-wise. Another thing that they have to think about is how does this relate to what we already know about stress? So what we know, for those of you who have come to Journal Club before or have taken some basic biology courses, corticosterone is very popular in what scientists look into in the study of stress. And it's involved in a lot of different parts of the stress response in the body. It's a steroid. But what does that have to do with what I just presented on? There's no overlap. I didn't talk about corticosterone, and then in corticosterone papers, they don't talk about Dicer1, typically, unless there's, whatever. But, so, how do these interact, and how do they relate? And if they don't relate, then have we fundamentally misunderstood stress and assumed that there's only one type of stress rather than two different roots of stress? And lastly, stress and anxiety, and yes?
What about GABA or dynorphin?
That is a fantastic question, and I'm really glad you asked that. I'm going to get back to you later on it. The last thing that I want to talk about is right now, anxiety and depression drugs are mainly out there to combat the symptoms of what these problems are causing. So what this could mean here is that what this research shows is there's a root to stress. There's a root to what's happening. And they might still have to find it, but it's important to know that now there's something that we could maybe combat at the root. We can combat at the base of the problem rather than just the top of it, which is really, really important and really, a good way to look at, maybe fixing the problem rather than just masking it. Yes?
But wouldn't that also interfere with the adrenal system and natural flight and fight? If we're targeting the root of stress entirely, then how do we just target chronic stress?
Well, if you were looking at it, it's not that the mice, if you look closely at it, it's not that the mice didn't have stress. The ones with the control, didn't have stress. It's just they had more stress without it. So it's not that we're trying to cut out stress, and obviously that's something they need to look into. That and along with other ways that upregulate these things, the other implications that these have. Because Wnt signaling pathway, when that's upregulated, the upregulation of this pathway and of aspects of this pathway have been linked with cancer. So they have to look into, can we knock out stress? Is knocking out stress, is getting rid of it a bad thing? Or are there different aspects of stress and we're just knocking down one part of it? This is just the beginning of a lot that they can look into with this. And I know that as Pingree students, we all feel stress, but we don't really know what it comes from. We only know, oh, I have a test. It's making me stressed. But what's actually, it's not the test going into your brain saying, oh, stress. It's your body's reacting to it. And this is a huge step in figuring out why you react the way you react. Yes?
Did they look at the stress pathway for the Bully-Meister?
I'm sorry?
The stress pathway for the Bully-Meister?
Yeah.
Because, I mean, it's conceivable that you would have a similar stress pathway that's resulting in different behavioral phenotypes, right?
This paper didn't look into that. There are a bunch of other papers out there that have been studying. This is really recent. In the past, they've looked into disheveled, which is another part of this pathway. It's another approach in this pathway. It's called disheveled.
Are all the vowels taken out or something?
It's like ESV or something like that. Yeah.
They're named after the fly phenotypes. They're named after what the fly acts like.
So there's been a lot of other research on it. This is the newest and most profound, and in my opinion, the most profound. But if you'd like, I can look into that and get back to you. Any other questions? Aaron?
Do you know why they use the bully mouse model to induce stress? Because I would think that like using live mice to induce stress in other live mice would be less consistent than like doing something negative.
Accelerated social defeat is actually used in a lot of papers. Bully mouse is pretty much an easier way to say it and a more basic way to explain it. But what they did was they subjected mice to social defeat. So mice somehow socially feel defeat. I am not going to try to explain that because I don't know it too well. This is a way that they use to induce stress consistently.
Okay.
I can't explain why or how, but I just know that it's not the first time they've used this method. That's all we have time for today. Any questions, please email me or come up to me in the halls. I'm more than happy to answer.